So for this video, I'm going to be discussing the origin of blood cell types and how to identify them. So all red blood cells, most white blood cells, and platelets are all produced within the bone marrow. Now, within the bone marrow, all these blood cells originate from the same type of stem cell, which is a hematopoietic stem cell. And this stem cell differentiates into two progenitor groups, the myeloid and the lymphoid. The lymphoid then can differentiate into natural killer cells, small lymphocytes, which are T cells and B cells. The myeloid class can differentiate into megakaryocytes, erythrocytes, mast cells, or myeloblasts, which the myeloblasts then can differentiate into basophils, neutrophils, eosinophils, and macrophages. So the first type of leukocytes or white blood cells is macrophages. These are some of the largest of the white blood cells. They are phagocytes, so they eat other cells that they perceive as harmful. They will eat anything that they think is, or they perceive as a pathogen or a foreign body, and then they will actually display that on the outside of their cell in these complexes called MHC class II molecules, in which they'll react with B cell and T cell receptors. The next three types of white blood cells are all granulocytes, eosinophils, neutrophils, and basophils. Granulocytes just means that they have granules inside them, like little packets of toxins that they will use against cells that they perceive as foreign or viral or anything. Eosinophils are sometimes phagocytic. Neutrophils are more phagocytic than eosinophils. Basophils release histamine, and they are involved in inflammatory response. The last type is a lymphocyte. There are three types of lymphocytes, which are B cells, T cells, and natural killer cells. All of the granulocytes have multiple nuclei, whereas the macrophage and the lymphocytes are mononuclear, so they only have one nucleus. So to talk a little bit about T cells. T cells do originate in bone marrow, but they move to the thymus gland to mature, and this is also where they will multiply and differentiate. And they can differentiate into T helper cells, T regulatory cells, cytotoxic T cells, memory T cells, or natural killer T cells. Natural killer T cells are not the same as natural killer cells. So in the thymus gland, this is where the T cells will meet the virgin B cells. The T helper cells specifically have a CD4 receptor, and that's a way to identify them. As well, T regulatory cells also have CD4 receptor. They also have a CD25, as opposed to cytotoxic T cells that have a CD8 receptor. So that's how to differentiate those. So like I said, T helper cells meet these B cells in the thymus gland, and they give them permission, so to say, to clone. So they'll help in the maturation of B cells. They give them permission to clone into plasma cells. These T helper cells, also called CD4 plus cells, since they have the CD4 receptor, they become activated and begin to differentiate when they're presented with antigens with molecules presented on with antigens presented on MHC class II molecules that are expressed on the surface of antigen producing cells like macrophages. Once they are activated, they secrete small cytokines, which are proteins that regulate and assist in the immune response. T regulatory cells, they regulate the immune system, regulate the immune response by suppressing T cells. Whereas T regulatory cells and T helper cells recognize MHC class II molecules. Cytotoxic T cells, on the other hand, recognize antigens presented on MHC class I molecules. When they come in contact with antigens on these MHC class I molecules, they will essentially ask them two questions. One, are you self? And two, are you carrying antigen that is self? If they are carrying antigen that is self, and they are self, they say, okay, you're fine. If they're not carrying antigen that is self, if they're carrying a foreign antigen, viral antigen, anything that they perceive as harmful, then they will lyse these cells. As you can see here, these are some drawings I did of the T helper cells, T regulatory cells, and cytotoxic T cell. A good way to differentiate these is the receptor. The T helper cells and the T regulatory cells both have CD4 receptors, as opposed to the cytotoxic T cell, it has a CD8 receptor. Memory T cells are another type of T cell. They may either carry the CD4 receptor or the CD8 receptor. These memory cells are important because they provide the immune system with a memory against pathogens that they have previously encountered. Natural killer T cells are another type of T cell. They are rare and they share properties of both T cells and natural killer cells. They are not the same as natural killer cells. They are involved in immunoregulation of other cells. They are able to recognize antigen classes other than MHC. And once they are activated by these other MHC class of molecules, they are able to perform essentially the same functions as the CD4 and CD8 cells, like cytotoxic T cells, T regulatory cells, memory cells, and T helper cells. Natural killer cells are large granular lymphocytes. They are present in blood. Natural killer cells make sure that all the cells they come in contact with are producing enough MHC class I molecules. Natural killer cells differ from cytotoxic T cells for this reason. Some infected cells don't want to get killed by the cytotoxic T cells, so they will stop producing the MHC class I molecules that the cytotoxic T cells will bind to and recognize as an antigen. So the natural killer cells will make sure that all the cells they come in contact with are producing enough MHC class I molecules. In this way, these cells cannot block apoptosis, because if natural killer cells see that they are not producing enough MHC class I molecules, then they will initiate programmed cell death.